Hello everyone. Good morning, good afternoon, and good evening. I'm happy to see that you are joining us today all over the world. Today we join forces with APC to be able to bring to you the webinar on solvent selection in pharmaceutical crystallization process development. My name is Carmen Guguta. I'm your host today from Technobis Crystallization Systems, and I have together with me today our presenter from APC, Dr. Marko Ukrainczyk. Just before, just before I will introduce Marko to all of you, I will like to share a bit of information with you about this webinar. It's being recorded, and I will make it available this evening, European time, or Monday morning to all of you. Depending on how time allows us today. You are being muted, but if you have questions, please feel free to write to us by using the chat function of this webinar, or just write to us directly at info@crystallizationsystems.com. You can write to us to this email address even after this webinar. And next week, together with Marko, we will try to answer all your questions. This webinar will last about 30 to 45 minutes. We will try not to make it too long for those of you that you are located now in Asia, and it's quite late in the afternoon, evening to you. I am actually happy to see that many of you are our customers. You are either a user of the Crystal 16 or a crystalline, or even both of them. So most of you know Technobis Crystallization Systems, but I see also a lot of new names. For those that do not know Technobis Crystallization Systems, we are a Dutch company located in the north of the Netherlands. And in our product portfolio, we have 3 products that we are marketing for solubility screening, crystallization, crystallization process optimization, and formulation related research. Crystal Breeder on your left side, the Crystal 16 and the crystalline instruments. We have more than 500 systems sold worldwide, mainly in pharma, agrochemicals, and fine chemicals. Relevant to this webinar today, the Crystal 16 and the crystalline instruments, they are multi-reactor systems that you can heat and cool in a controlled manner. The Crystal 16 is your instrument at 1 milliliter scale. It is the instrument for solubility screening, metastable zone with determination, phase diagrams, if you want to construct phase diagrams, determine induction time type of measurements, solvent screening, any type of solid state type of screening, polymorph, salt, co-crystallization, even amorphous dispersions, deracemization type of experiments. Crystal 16 is your instrument to use. Crystalline instrument is working at 5 milliliter scale. With the camera, it comes with the camera technology that works exactly like a microscope. It helps you to develop or optimize your crystallization process, your formulation process. You are able to determine particle size distribution, control the habit, the form. It is quite flexible in the gadget that it comes with, and with the camera technology, or with Raman. If you have a Raman spectrometer in your lab, you can easily connect it to this instrument and follow polymorphic transformations or even chemical reactions while you are performing the experiments. If at the end of this webinar you think, I need a Crystal 16 or I need a crystalline in my lab, I'm pretty sure one of my colleagues can make something available by the end of this year to have one of these instruments in your lab. Just write to us at info@crystallizationsystems.com or contact us via the website. I'm pretty sure my colleagues from sales will be happy to help you further from this. And now I would actually like to introduce our speaker today, Dr. Marko Ukrainczyk. He is currently the senior scientist, technical lead of the small molecule crystallization process development team at APC in Dublin. He has a PhD in physical chemistry, and he is a chemical engineer. I'm sorry, Marko. Prior to joining APC, he has done his postdoc at SSSP Center in Ireland, together with Technical University of Munich and Karlsruhe Institute of Technology, Germany, specializing in crystallization. As a chemical engineer and physical chemist, he combines computational and experimental techniques in his work, mainly focusing on thermodynamic and kinetic aspects of crystallization as an efficient synergy for improved process design aimed to speed up the delivery of life-changing medicines. Marko, thank you very much for sharing with you your knowledge and some case studies today. I am making you now a presenter, and I think now you will be able to share your screen with everybody.
Thank you. Thank you, Carmen. I'd like to thank you for this opportunity to present here today. So I'm Marko and coming from APC, from Dublin. And just a minute to clean the screen here. So APC is a pharmaceutical company. It's an R&D company located here in Dublin. It's just a half an hour walk from Irish Sea coast, and with lab and office spaces just overlooking the Wicklow Mountains. So it's a very nice place to work in. It's APC is involved in process development. So we are involved in reaction optimization, crystallization, then all the way across distillation, filtration, and drying to formulation platforms. We are focused on both.
Marko, yes? Just one second. I do not see your screen.
It is. I am projecting it. Just a second. Let me see.
Yes.
You see now?
Perfect.
Okay. So we are focused as APC on both early stage and late stage development, and it is for both small molecules and large biomolecules. And here today, I like to present a topic on the solvent selection. So it's our solvent selection platform, which is done together in partnership with Janssen. So it's a crystallization technology unit in Beersa, Belgium, with Ronnie Weidenschot and Alain Colas. So I'm happy to present the case studies which we work together. But before I go to the case studies, I would first like to introduce you to the topic, then present a little bit on the in silico predictive modeling, which is our integral part of our early development, crystallization development. Yes, and I will also introduce you into the workflow, which combines in silico screen and smart experimentation. And then, as I said, I will demonstrate our workflow on two case studies. So it's two proprietary API molecules. One is an interesting example of antisolvent crystallization, and another one of a cooling crystallization. So this is what I planned for today. So selecting an appropriate solvent. This is the cornerstone of a good crystallization development, and the solvent system from which API is crystallized influences solubility. Just let me see if I can get the pointer. Yeah, so solubility, growth and nucleation kinetics, impurity rejection, polymorph control, solvation and oiling propensity, and crystal morphology. So as you can see, it's very important to select the right solvent. But despite this importance of this choice, usually early development solvents are often chosen quickly with only the impact of product yield considered. And this can lead to problems or processability issues, or for example, purity issues in later phases, either during the scale-up or optimization phase, or even when it comes to the manufacturing, when the material is being delivered to either clinic or market. So our developed solvent selection platform is based on evaluating 12 criteria. So we are going all the way from yield, productivity, seedability. So all these criteria which are pointed here, and also the anti-solvent efficiency, as well as safety. So it's we are also considering safety and environmental choices. So, and we are doing that by using in silico predictive modeling and the smart experimentation. And I just want to maybe compare it with other approaches, which is, for example, more traditional and quite expensive and time-consuming brute force approach. So, but we are what I would like to claim here that we can do with our approach same or less time. So we can reduce the early development timeframes and also the risk. Regarding the time, as I said, less or same or less time than traditional brute force screening. And regarding the risk, I believe that the approach which I'm presenting today, it dramatically improves the likelihood of selecting a fit for purpose solvent for crystallization processes. So we are basing our approach on quality by design principles, and here quality by design principle we are basing on in silico modeling. So on this slide, I would like to tell you more about it. So in the case of crystallization, of course, everything starts with the most important physical property, which is the solubility. And unlike in late stage crystallization development, so we have it here, where accurate regression models are usually employed to map the full solubility design space, so usually as a function of temperature, solvent composition, and also maybe pH, based on time-consuming experiments. In the early phase, so what we have here, during the solvent screen, those predictive models prove to be extremely useful and important, especially for screening of large number of solvents and solvent mixture. So in this case, the accuracy compared to the empirical model is lower, but still satisfying to guide and narrow down the experimental work. As an example, here I'm showing the regressed unifact, which is implemented in DynoChem. So the solubility prediction is possible based on provided molecular structure, but also on experimental solubility data and some DSC data, which are required as a minimum. If we go even further to the first principle methods, so which are physics-based, all the way to molecular electron scale, so like, for example, quantum mechanical calculations. So in this case, I want to emphasize only molecular structure is required. So as we can see, this makes them very powerful for early stage development, because usually in early stage, there is no data much available, and also the material, there is usually no sufficient material available yet. So the solubility and the solvent screen can be performed just ranking the solvents from, for example, from the potential very dissolving solvents to anti-solvents, as well as solvent mixtures. Here for this type of calculations, at the moment I covered the solubility, but I also wanted to emphasize if the molecular structure of impurities are provided. So the solvents can also be screened for impurity purge without the need, for example, to have any impurities isolated for experimental work, which we found in EPIC it's usually the case. Yeah, we know the impurity structure, but very often we don't have standard materials of isolated impurities. I also would like to highlight that we can also screen for solvate propensity and co-crystal, as well as oiling propensity, and even going further, crystal shape and polymorphism. So for example, if you have a unit cell, so crystallographic data, crystal shape, and also the polymorphic stability can be studied. So now when I introduce you to the methods which we are heavily using for the crystallization development, I would maybe like to go through the time now. So now in the different direction. So I would like to start the way we are doing usually in APC. So we start first with early development. So it's the first principle methods, because as I said, there is no or little material available, and there is no data. But once we generate and select a solvent and we go to the lab and do experiments, then once the solubility experimented data are generated, we then input that in the UNIFAC models. So we are using these platforms. And then generating even more data, experimental data, we are ending up creating the full map, which then is used for the crystallization design. So for the first principle models, I would like to introduce you what platforms we are using. So first is quantum mechanical calculation. This is density functional theory based calculations, where solubility can be extracted based on calculating the polarity profiles of APIs and also of solvents. And by creating and solving the electron density of API and solvent, this can be used to derive chemical potentials of API in a solution. So here, this is so from this we can calculate relative solubilities, and it is the calculation is based first. You can estimate the combinatorial part, which is the difference between size and shape between solvent and API, and also it calculates. If you look more from the statistical thermodynamics, it's this residual enthalpy, or it's actually the interaction contribution, which accounts for example, hydrogen bond, electrostatics, or van der Waals interactions, which are introduced by inserting the API molecule inside the solvent. Also, solvent propensity can be calculated by calculating enthalpy of mixing. So this is now based on the interactions, and it can be calculated as a difference between when you have a mixture of solvent and API, and the difference as only API and only solvent. So this is the excess enthalpy of mixing. And for example, if you have solvates where there is a high chance of hydrogen bonding, the interaction will be highly negative. And this can be used for during the screening for the identification of potential unwanted solvates. As a second method, we are also employing the molecular dynamics. So this is now based on molecular mechanics, instead what I previously introduced on electron density. And here atoms have point charges. The bonds are, for example, represented by spring potential, van der Waals interaction, for example, by Lennard-Jones potential. And we use such methods to explore, for example, conformational space. So conformational space of a molecule, it can be explored in a vacuum, but also we can explore it in, for example, different solvents. And another thing we have also is lattice energy calculations. And as I will also demonstrate later, it's also we can predict the crystal shape. Crystal shape predictions we found quite useful because, for example, different polymorphs usually they exhibit different crystal shape, and just knowing what shape looks for different polymorphs, it's quite easy, for example, identify them by microscope. Our solvent workflow starts with a large high throughput computational screen. And usually we screen up to 70 solvents. So we identify the most suitable solvents in terms of solubility, so yield, and also exclude those solvents which shows, for example, high propensity for unwanted hydrates or solvates. Starting from a set of 70 solvents, we identify typically eight solvents, which we go forward to the experimental validation. So for the experimental validation, we usually do 2-point solubility. And in this kind of experiments, so these experiments are done in crystal 16 usually. And here we don't only look for solubility, but also for potential degradation. So for the purity, and as well, we always isolate the solids and we look for the form. So 2-point solubility is it's two temperatures, low and high temperature. And after this, we are narrowing down even further. So usually to 2 or 3 solvents, which we then perform a small-scale crystallization experiment. And this is usually done in crystalline. So here, we are looking for metastable zone width, form, purity, crystal shape, yield, and productivity. So just to give you maybe a bit more detail. So starting with the computational screen. So we select based on solubility and solvent class, and maybe a chance to form a propensity of a solvent. We select 8 solvents. Then we go forward with 2-point solubility. And here I want to say, if the solubility data shows that it's possible to get the yield higher than 75%, in this case we are proceeding with the screening crystallizations, which are cooling crystallizations. And usually we do it unseeded. Why unseeded? Well, because we want to also in the same run, we want to see by slowly cooling it, what is the metastable zone width. After we cool it down, we are keeping it at 1 temperature for aging, and then we isolate. So then we are looking what is the form we are isolating, and what is the purity of the solid and also of the mother liquor, and what is the morphology, and finally we want to confirm the yield. Is it exactly what we are seeing with the solubility? And after that, we select usually one option, which then goes for the process definition. The process definition, we usually go to the 100 mil or 400 mils automated reactors, which are fully equipped with, for example, particle track, so FBRM, and also IR. So here we have a decision tree if from solubility data, if the yield is not possible to achieve just by one single solvent higher than 75%. So then there is no chance to go most probably for cooling crystallization, but we need to select an antisolvent, which will further improve the yield. So usually here we are limiting down to 2 dissolving solvents, because coming into binary mixtures here, having additional 6 antisolvents, that of course increases the solvents which needs to be screened. But here I would like to emphasize, especially the computational screen is quite powerful, because we can screen many conditions before selecting only few interesting points where which we validate with the experiments. So we select 6 antisolvents from the previously done computational screen on neat solvents and antisolvents, and usually the selection of 7 relevant antisolvents is done on miscibility, and also we want to select green antisolvents. Yeah. So once here we are creating the so it's a solubility as a function of solvent composition, starting from 0 all the way to 50 weight percent, or even to 100 weight percent. And here we are looking into the detail of how efficient is the antisolvent. Is there any co-solvency effect, or is there solvation synergistic effect? So based on that, we are proceeding with experimental, so isothermal solubility, and here usually we pick up only few experimental points. If there is a synergistic maximum, we have experience usually it is around 50%. So this is what we want to check. Is it really the synergistic maximum? We want to validate if the computational screen is really predicting that synergistic maximum, that it's really happening. And in case there is a significant drop, so the antisolvent is quite efficient, then we go, for example, to test at 50%, is it really reducing the solubility drastically? So after that, we are selecting 2 solvents, and we are going to screening crystallizations. Again, we are doing unseeded crystallization. This time it can be cooling, just first part, but then definitely we are adding antisolvent. So first we're adding antisolvent. We can cool it down, or we can just proceed with the simple precipitation method. In this case, we are also noting how much time does it take that it nucleates, and we are also testing for form, morphology, and yield. And then we go to the process definition method. So here I also emphasize if the yield is higher than 75%, but in case metastable zone width is poor, so we have a poor nucleator API. So then we are also considering to introduce the antisolvent before it can be nucleated and crystallized. Okay, so I covered all the workflow. So now I'm switching to the case study. And our first case study is an API. And it's a really API with a very high melting point, so 215 centigrade. So here you can already see just by looking at the melting point that the compound probably will be poorly soluble in most of the solvents, since API is quite stable in crystal lattice and doesn't like to disintegrate and dissolve and mix with the solvent. So, and before going to the solvent selection, I just want to briefly introduce you maybe to the background of the compound. So in this case, we have two polymorphs, and they are enantiotropically related to each other. So the desired one is form 1, and that's the one which is the Janssen went for the commercial form, and it's only stable below 50 centigrades. When we got two forms here in APC, we performed our DSC analysis as well as Raman, and we found out it can be clearly distinguished not only by DSC and Raman, which it's always a benefit because especially in early development, you can work with smaller quantities. We also had an indication that it has a high solvate formation propensity. And in terms of particle attributes, there was no specific requirement for output particle size distribution, only that it filters well. And this is because there was a spray drying technology on the formulation. So in terms of purity, there was a lot of problems there because first we developed the crystallization using it's pure material. But then once we started to test our developed crystallization with the crude input material, the crystallization didn't behave the same. And also the crude materials, there was a lot of variability. And so there was already identified impurities, but we also in APC identified that there are also some unidentified impurities. And I will tell this a bit more detailed later, where we actually managed to identify those impurities by Raman, just by looking at the fluorescence. So these were most probably fluorescence impurities on the ppm levels. So here we performed, as I mentioned, the computational solvent screen. In this case, it was about 40 solvents. And also we included exotic solvents, so high boilers like NMP, DMPU, TMI, NBP, just because we knew it probably won't dissolve in most of the solvents, which turns out to be the case. And in parallel with the solubility, we also performed the solvent formation propensity. So we calculated that enthalpy of mixing is highly negative for all those high boiler solvents, which was a red flag because it showed potentially, there will be problems with solvates. And once we performed our solubility data, so these are the solvents which we went for. Just because although we knew there might be a problem with forming the solvates, we didn't have more choices because all the rest solvents didn't give much solubility. And especially we couldn't afford to go to higher temperature because we wanted to isolate the form 1, which is stable only below 50 degrees. So we didn't want to risk the formation of the undesired form. So here, doing the solubility, experimental solubility, we can see that the ranking order was right. So this we computed the solubility, relative solubility based on solvation free energy. So we did a non-iterative method. So by and here the solvents are ranked, and the same order we can see in the with the experiments. In these cases where I don't have the values, it's either because the solubilities was too high, or actually in this case was happening that the solvate mass formed. So there was no mother liquid sample for solubility, which already proved to be problematic and probably showing to solvate formation. So all solids were analyzed by DSC and Raman, and here I'm showing Raman, which nicely confirmed actually that we are forming a solvate. So usually for solvate there is a shift, which we can also see here. In this case, there was also a doublet forming, but definitely just by looking in the Raman, we didn't had the form 1 or form 2, we had a new form, which just by looking in DSC, we confirmed it's a solvate. So having all of this data together, we actually went for the only solvent, which was the option, which is a THF, because it has a high solubility and it was giving the right form. So having a THF as a dissolving solvent, we definitely needed to find a proper antisolvent so we can crystallize our material. And here we performed a binary mixture solvent screen, antisolvent screen, and we considered from water all the way to heptane and methyl cyclohexane and toluene. And we found out that water gave a quite high synergistic effect, which was proven also by experiments. And we also found out computationally with the red here curve that heptane is the most efficient antisolvent, which was first confirmed by only this point at 58%. And then later when we identified our solvent system, so THF heptane system, we also built up the solubility curve. And just by looking in the solubility, 96% yield is achievable, and also with a high throughput of 12 grams per 100 gram. So this was put forward to the process definition, and based on our solubility data, we designed our crystallization. So first, we are starting. So it's an isothermal antisolvent crystallization. But just to increase the throughput, we are actually dissolving our material to just below 50 degrees, cooling it down. Then we are adding antisolvent, so making the solution supersaturated. We are seeding, and we can see from ABRM trends, there is a secondary nucleation, which equilibrates after the aging time. Also, IR shows that the solution is nicely desupersaturated. And after that, we are adding the final dose of an antisolvent, which there was no secondary nucleation, unwanted secondary nucleation happening, which turns out to be a very nice crystallization design. Yeah. So this was optimized and fully scaled up to 12 liters. So this was done at our colleagues in CTU, and during the scale up, there was a right form, there was a good filterability, and there was a low residual THF content. So a good process. I just want to highlight, once we switched to crude material, there was a problem here after the seeding. So the material didn't want to nucleate, or it nucleated a bit later. And in only one case, we found out that actually we got the undesired form 2. which was probably induced by impurities. So that was a problem. And just by analyzing crude materials, which we got here in APC, we found out that Raman, the baseline is quite flat for pure materials. But when we got the first crude material, it has a fluorescence and two peaks, which we calculated, which correspond. Yeah, we were thinking maybe it might be even two different impurities, but we weren't sure. But once our colleagues at in Janssen, they changed the crude step, and they managed, they said, to remove the impurities. So we got another crude material, which was performing better. And after recording the Raman spectrum, we actually found out it seems like that they managed to remove one impurity, but that the one was still there. And here I just wanted to highlight that the power of Raman. So it was able to detect fluorescence impurity, which were not been able to detect by UPLC. So, and the quantities was ppm level. But it's interesting because even though it's a ppm level, they even so low concentrations, they've been able to affect the crystallization. So in terms of prolonging the nucleation time and even having troubles with generating the right form throughout the process. So I'm switching now to the second study. So the second case study was, we have an API and the melting point was significantly lower. So it's 126 centigrade. In this case, we also got two forms, form 1 and form 3. Form 1 is the most stable. And it was also clearly distinguishable by DSC and Raman. Regarding the purity, API was unstable at elevated temperatures. And here the focus of the project was actually in terms of solvent selection. We wanted to select a suitable solvent, which will be then, we will use it so the solvent system can be used for further kinetic assessment for continuous crystallization. So it was a batch and steady state operation where we wanted to assess is it API suitable for MSMPR reaction. This work is done and presented in Orlando by Gladys, and I won't go into details, but I will show you how the solvent was selected. So in this case, we started with our computational work, and because the melting point was quite low, and just looking into data, when we computed some of the data in comparison to some limited supply data, very crude experimental data, we found quite a good match. So we got confident, okay, let's go for this time for absolute values. So we were using melting point and enthalpy of fusion as a reference. And in our software, we were able this time to calculate absolute values, not only for room temperature, but also at 50 degrees. So in this case, we were selecting the solvent in silico. And our selection criteria was, okay, let's go for cooling crystallization. So this is always preferred. And let's find a solvent with high yield and throughput. And so we were looking at the room temperature solubility data. Is low, that the slope of the solubility curve is high, and then there is a high solubility at 50 degrees. And we identified a number of solvents. So isopropanol, MEK, isopropyl acetate, and anisole. So with this we went further to the experimental validation. And in this case, we were using isothermal slurry method. And when we were isolating mother liquors and we were checking the purity, 3 solvents were ruled out because we found an impurity in those solvents at higher temperature. So then just by analyzing the solubilities, we found out indeed that isopropanol, anisole, and isopropyl acetate are good solvents. But out of those 3 potential solvents, actually isopropanol we went for isopropanol just because there was a very high solubility at 48 degrees, which assured that we will have a high throughput as well as yield. So this solubility is at room temperature, so 20 degrees. But we found out that even below 15, if we lower down temperature to 0 degree, we can get even higher yield. And with that, we went still with keeping. So we selected isopropanol as a runner, but still we were keeping additional 2 solvents just to check what is happening while we are crystallizing in all those 3 solvents. So here I am showing the crystalline, which we find it very useful for our screening purposes when we do our proof of concept crystallization experiments. Especially when we use overhead stirrer, we can also look in the morphology. And here I'm plotting the trends which crystalline is giving us. So it's a temperature and also turbidity. So we are first starting with slow cooling, and we can observe where the compound is nucleating. So we are noting down metastable zone width, and after that there is an aging period, and then we are cooling further to 10 degrees, and then again waiting period, and then we isolate. I want to say here maybe for crystalline it's nice the camera which is embedded in the crystalline. We can see the shape during the nucleation. Unfortunately, later it's not possible to see just because it's too many particles there. But at the end, this is the microscope, which is the morphology analyzed by microscope. Yeah, and we can see that the morphology is rod-like, and also we observed what is the concentration and what is the yield. So we got 88% yield for isopropanol. There is a form 1 which is desired, and there is no impurities detected in the mother liquor. So when we presented this to Janssen, to clients, this was perfect, nice, but still having a yield of 88% is not ideal. So it's more desirable to increase the yield. So in this case, that's why we decided, okay, let's go and screen for antisolvents, which can be added at the end of the cooling crystallization, so just to get a better yield. And we did first computational screen. All of the antisolvents gave a bit of synergistic effect. So there was always not so efficient antisolvent at up to 30%, but then later we were getting quite nice, especially for heptane. And this is the antisolvent we selected. And then we mapped not only at the different experiment and not only different solvent compositions, but also different temperatures. And here it was possible to increase the yield from 88% by adding antisolvent to 94%. The only maybe, let's say, downside was now that we are reducing the throughput by half, just because we need to add 58% of a heptane. And with this system, we went through the process definition. Even though we were already looking into the action times and metastable zone width at the crystalline scale, we are always repeating this also at for example, 400 mil scale. It's maybe the best because those nucleation induction times are more reliable and representative of what will happen on the industrial scale. And just by noting what is the metastable zone width, so based on that we chose what is the seeding point. So 30 degrees in this case, and after cooling down, there was unwanted nucleation. And after cooling down, we were adding heptane up to 50% just to get the yield. And I think this is also very good run, this is only process definition assessment. So first run, and of course it can be still optimized. but it gave a high yield, no solvation, and the very high throughput, and the particle morphology was acceptable. So now, when I mentioned crystal shape, I want to maybe go more detail about that. So we are using crystal shape prediction also here in APC, and this is crystal shape prediction is an active area of research. So different modeling approaches exist. So, for example, evolving from geometric, energetic, and also to the mechanistic, like spiral growth or Monte Carlo simulation. And here the prediction accuracy increases gradually, as well as complexity. And what we found most useful, and it's most widely used, it's an attachment energy method. So it belongs to the energetic method. And the reason why it's widely used is because it's simple calculations and relatively reliable accuracy. So it's quite valuable for screening purposes. So those methods, the morphology is built based on calculation that the growth of a particular phases is proportional to the lattice energy, actually energy of the slice. When the slice is put into the bulk crystal, and that energy is released, and it's done for a particular crystal phases. So, and the slowest growing phases becomes the morphologically the most important. So as we can see here on this slide, we have here predicted morphologies, and they are very much in line with the experimental observed morphologies. So I want to emphasize, even though this model here doesn't consider the solvent environmental, but it's still good, and probably due to the fact that the shape in most cases is indeed predominantly controlled by internal interaction within the crystals rather than with the solvent environment. And this, I think, we found in APC especially the case holds, and it holds true for molecules which with dominant van der Waals forces. So those kind of models, once the shape model are generated, are quite useful because we can look into the surface chemistry of individual crystal faces, and we can think about what solvent might be, for example, best. If we have a needle-like shape, maybe they can help to suppress of those fast-growing phases, for example. And this is an example where individual crystal phases were simulated, and in this case, it's in interaction with the solvent environment, and the shape is constructed. Yes, again, based on the attachment energy, but in this case, it's attachment energy. It's a modified attachment energy because we are also considering the solvent environment. So we have here three different settings: vacuum, where there is no external interactions on the left-hand side. Then it's less polar environment in the middle, and there is a more polar environment. And we can see that the aspect ratio in this case is affected, and that the polar solvent most probably is inhibiting the growth of fast-growing phases. And this was also in agreement with the experiment. We can see here needles for less polar solvent and quite so with the less aspect ratio and quite compact crystals in the case of more solvent environment. So with this, I would like to summarize what I presented. So I hope you were enjoying the presentation, and I hope I managed to demonstrate our workflows for early phase crystallization development. So I presented one case study with isothermal antisolvent crystallization and the other one with the cooling crystallization. And I hope you got interested in the methodology which we are using for the solvent selection. So which we based on in silico predictive modeling and also followed by smart experimentation. And this is for smart experimentation. we are using parallel reactors, so crystal 16 crystalline. And I also hope I managed to demonstrate that also the Raman is quite useful for solvates and impurity identification, and as well as the form. And so in APC, our strategy utilizes advancing in thermodynamic modeling and to minimize development timeframes, and also to maximize the value of the choice made in the early stage development. And we hope when we get the project in APC that phase 1 crystallization can be successful right from the start all the way to the scale up and to the manufacturing, as it was demonstrated during the talk. So thank you. I'm open for questions.
Thank you very much, Marko. A good presentation and all this information that you have shared with us, and also to Janssen that allowed you to present these nice case studies with us. You have already shared with you your experience also with our Crystal 16 and crystalline instruments. Can you share with us any other applications? Because I'm pretty sure that you use these instruments for with more applications in mind. Is it anything that you can think about to share with our viewers today?
Yes. So what we found very useful, it's not only for small-scale crystallization experiments, but also very often we perform, for example, competitive slurry trials, and also to map up the thermodynamic phase diagrams. But also, what I found very useful, crystalline especially, it's for kinetic, to map up the kinetic phase diagrams. So these are usually experiments done in crystalline, where we seed usually a mix of the form, and then we just want to see what form will crystallize. So not wait for the thermodynamic equilibrium, but just to see what will crystallize after the seeding. After, for example, after 3 hours. So this is what I found very useful. Also, as I mentioned, it's for solubility, for polythermal solubility. So there is many reasons why to go for crystalline. And maybe the most is just because it's on a small scale, so we don't need to use much material, but it's also quite handy and fast. So we can, for example, when we perform one run on 100 mil, I think we can perform much more runs on the crystalline, for example, 3 or 4. So it's just faster. Yeah.
Okay, thank you, Marko. We have quite some questions, a lot of questions, and a lot of also good notes, nice notes. Thank you, all of you, that you have experienced nicely. You found this presentation of Marko interesting. I'm trying to read now some of the questions. One of the questions from Joe Gao. What is your assessment on accuracy from the solubility prediction versus experimental results?
What did you mention at the beginning? What is, I missed this part.
What is your assessment on accuracy from the solubility prediction versus the experimental?
Okay, so that's a good question. Yeah, we found sometimes works very, very well, especially if the melting point of the material API is low. We could probably look then at absolute values. In some cases, it works very well. In some cases, there is a bit of drift, and especially if you go to high melting points, which is probably due to not considering the heat capacities. Yeah, but in general, what I found actually for screening purposes, it's not much focused on the really absolute values. So we really use the tool to screen and to propose a suitable solvent, which we can then really get the accurate value just by doing the experiments. So
yeah,
I hope I answered. Yeah.
A few of our attendees, they are asking on the same topic, how long does it take to do the computational screening of 70 solvents?
So the time-consuming part is actually that quantum mechanical calculations, which usually for the molecules takes about four hours, or it can take longer. We had API molecules which took the whole night, but this is just to build that quantum mechanical calculation, and then later actually the calculations are quite fast, so it can take 20 minutes to 1 hour. If you do iterative approach, where you want to include also the concentration of API, I found out also when you calculate high solubility, that it can take a while, just probably to converge. So I had cases where it took half a day.
Okay. Another question from Marko, but this time Marko from BI, Sant'Agostino. He's asking you, which precision in the computed solubility data do you usually get? Can you comment on charged molecules like salts?
So salts are more difficult to predict the solubility, but we were modeling salts in some of the cases, and it worked reasonably well. But there was also some cases where it just didn't work much. So I can just comment, yeah, the salts are a bit more difficult, and sometimes it can work, yeah.
Okay. On slide 11, Nicholas Rim is asking, can you explain why Form 1 has 2 DSC peaks while Form 2 has 1 peak?
Yes. So where did I hit that? So here. No. On the
11.
First. So
yeah,
the green one, that's the desired. I hope I'm not. Yes, that's the Form 1, and it has 2 peaks. Yes, you're right. So first it melts, but then it recrystallizes to Form 2. So that means.
And then it melts Form 2, most probably.
Yes, but these are enantiotropically related, so it's a more complex.
Oh, okay.
So that means at a higher temperature, actually Form 2 becomes the most stable. And this can also be seen by just if you look at the enthalpies, because the Form 1 melts first, but it has a smaller, lower enthalpy, and then, but definitely at higher temperatures, Form 2 becomes more stable. That's why it converts into the Form 2. Yes.
Okay. Another question, how did you control polymorphism during crystallization in case study 1? Once you dissolve the API, how did you manage to obtain Form 1 and not Form 2?
So in this case, we did competitive slurry trials just to make sure that below 50 degrees and the solvent composition, which we are working, that we are always isolating Form 1, and all showed that we will isolate Form 1. And if you look here, if we are below 50 degrees, it seems like, it will be the case, which we confirmed, even though we already got that information from Janssen. We confirmed that by performing the competitive slurry trials. And and throughout the crystallization, not only at the isolation point, but throughout only Form 1 we were getting.
Okay, there are a lot of questions. I will make them all available to Marko, and somewhere next week he will come back to all of you. I will take just one more question. Well, actually, this is one good for APC. Very interesting, Marko. Do you do contract crystallization research? I think the answer is yes, definitely. You will have also the contacts of APC shortly after this email, after this webinar. But one more question. Great presentation. Thank you. This is from Edwin Torres Cuevas. Yes?
Can I just comment?
Yes.
Yeah, thank you for your comment. I just want to say maybe just a few weeks back, we actually created a new website. So it's approcess.com. And please go there because the website is created for the process scientists. There is a lot of resources there. So please visit.
Great. Thank you for this. One more great presentation. Thank you. I have a question about the Cosmotherm calculations. Earlier you said that because you got a good mesh, you were able to consider the DH results quantitatively accurate. What does a good mesh mean, and how do you compare it to a not good one?
So as I said, for screening purpose, I think if we select the right solvent, that's good enough. So the in silicon screening gave us isopropanol as the best. And then when we explored not only, so we explored number of solvents, and actually we got quite similar results. So there was a low solubility at 20 degrees, but a very high solubility at 50 degrees. This was predicted by Cosmotherm and also confirmed by experimental results. So I think that's a good match. If you are asking me about the accuracy, yeah, usually there is a bit of our experience is about like maybe even 50, 100%, but I think this is still good enough for screening purposes.
Okay, Marko, thank you very much. As I mentioned, there are quite a lot of questions, a lot of good words for you. Thank you for making this presentation available and share it with so many people. Thank you all for attending. I will make this recording available on our website this evening. It will be there, but most probably an email with the link to the recording you'll receive somewhere next week on Monday. I'm hoping that, we will also be able to share the slides with you. If you have any other questions, please do not hesitate to send us an email at info@crystallizationsystems.com or at apc@approcess.com. We will be happy to further help you. Stay safe and healthy during these challenging times, and hope to see you soon to our next webinar in November. Thank you all. Thank you, Marko. Thank you, APC.
Thank you. Thank you. Thank you.
Thank you. Bye.